For our final project, we went with infectious diseases. We tested bathroom doors at the MSU Extension office. Our hypothesis is that the doors exiting the bathrooms will be cleaner because people are supposed to wash their hands when they exit, so therefore they will be cleaner.
The tryptic soy agar, or Tsa Agar, contains glucose and amino acids for the growth of most microorganisms. It will not select for gram-positive or gram-negative because both groups will grow on this agar. It basically just shows us that we have a particular bacteria growing, but not what type of bacteria it is. All of our TSA plates had growth with a range of 2 to 4 plus on the door handles going into the restrooms on each floor, as well as a range of 4 plus on the handles going out of the restrooms. The size of the colonies on our agar plates were dependent on how quickly the bacteria types replicate. Some bacteria replicate at a slower rate while others replicate rapidly. By knowing the replication rate, you can eliminate particular bacteria from your hypothesis. We did a catalyst test using hydrogen peroxide to determine if we had Staphylococcus aureus or Staphylococcus epidermidis. We used the hydrogen peroxide to see if our sample reacted when we combined the sample with peroxide. We had a positive reaction out of each TSA plate except for floor 3, sample 1 going out. We are uncertain as to the reason for the lack of this reaction on only the one door handle. If we used the Tsa Agar to determine what our sample was, we wouldn't have eliminated any of the possibilities with the catalyst test.
McConkey agar, MAC for short, is a selective agar that selects for gram-negative bacteria. It can also help us group bacteria based on the ability to ferment lactose. If they can ferment lactose, the bacteria colonies will turn a pink color. If they cannot, they remain colorless. Our results had growth on only 2 plates, exiting floors 2 and 3, sample 1. Both of the samples had colorless colonies, therefore they do not ferment lactose. Salmonella was most likely the type of bacteria that is on our MAC plates. We arrived at that conclusion using the following reasoning. E. coli ferments lactose, and Streptococcus pyogenes, Staphylococcus epidermidis, and Staphylococcus aureus are all gram-positive.
Mannitol salt agar, or MSA, is a very selective agar which contains a very small percentage of salt to select gram-positive bacteria, like Staphylococci. MSA also contains mannitol sugar and phenol red, which select for organisms that like to ferment mannitol. If the organism is able to utilize mannitol as an energy source, the agar will turn a light yellow. None of our plates turned yellow. All of our samples taken from the door handles on the outside of the bathroom showed a 1 for growth, except for 2 doors. Except for 2, none of our samples taken from the door handles on the inside of the bathrooms leading out of the hall showed growth. In reality, at least a couple of these black sheep were probably contamination. However, if we ignore this fact, we can still make a few conclusions about what grew on our plates. Based on the MSA plates, the bacteria that grew could not be Staphylococcus aureus because it did not ferment mannitol. Thus, the only thing we could have growing on it would be Staphylococcus epidermidis. Staphylococcus epidermidis is a catalase positive that produces normal flora on the skin and forms biofilm and adheres to medical devices. Some of the symptoms that this disease produces are infection of indwelling medical devices such as prosthetic joints or catheters. An example of a clinical case is 10 days after undergoing chemotherapy, a treatment for a cancerous man develops fever. Well, on examination, the patient has tenderness at the insert site of the catheter. Blood cultures are positive for both bacteria. The treatment for this would be to remove the original catheter and put the patient on antibiotics.
For our conclusion, we came up with that the exiting doors are dirtier than the entrance doors to the bathrooms on floors 3 and 2. We found salmonella on the exiting handles of the doors.
And also, thank you to all the MSU professors
For all your help and time with the BioScience project.
And thanks for watching our video.
together.
Okay, any questions?
You have paper towels or electric air dryers in the bathrooms? What?
Paper towels.
Paper towels. Okay.
Paper towels.
Yep. Thank you.
The question was a regular office or was it in the courthouse?
Can you say it again?
Is it in a regular office or is it in the courthouse? Did they ask a question?
We will never know.
That's Ravalli.
Yeah. Any other questions, you guys?
Did you really check the outside of the outdoor? Somebody said.
Of the bathroom.
No.
Inside of the outdoor.
Both sides.
Most of the bathrooms are
The inside of the bathroom door, the outside of the bathroom door.
Oh, I see. Just one door.
What was your conclusion? Because the inside was had more stuff growing, right? That was the opposite of your hypothesis.
Yes.
Answer. She didn't hear me.
What's that?
What you found was the opposite of your hypothesis. Is that right?
Yeah.
And so did you have a conclusion for why that might be?
People don't wash their hands when they leave.
But the
We're not well enough here.
But the alternative, I've heard an alternative theory anyway. I've heard that you have to wash your hands for a long time. You have to be able to sing happy birthday to you all the way through while you wash your hands. Or what can happen is if you just put a little bit of water on and brush them around, even a little bit of soap, then you're solubilizing the bacteria that are on the hand onto the doorknob. Whereas when the people come in to the bathroom, their hands are dry. And so maybe they're getting less bacteria on the outside just because the bacteria aren't soluble.
Maybe you need to put a webcam in the bathroom to see people wash their hands.
That's gonna go over well. That's what I do.
Did you guys see the question from Titan about how many people went in and out of the office, do you think?
In and out of the bathroom?
Or in and out of the bathroom, I mean? Yeah.
What are units?
These were all male bathrooms?
Yeah.
All of them.
No women are dirtier people.
We rest
I don't know if that's the hypothesis.
Now, were these like single stalls, or was it where you went to the bathroom? There were several options, for lack of a better word.
There was
Only one really.
I see.
There's a urinal and a commode at the end of the restroom. Or the one on the first floor, which apparently is.
Well done. I really enjoyed the video. You guys did a great job. That was awesome.
It was a great video. Thanks for hanging in there to play it for us.